Hello, everyone, and welcome to today's webinar. Discover how single-cell RNA sequencing provides deeper insights into lung cancer biology. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Illumina and 10x Genomics. To learn more about our sponsors, please visit illumina.com and 10xgenomics.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers: Bernard Thienpont, Assistant Professor, Faculty of Medicine, KULeuven; Annika Branting, Regional Marketing Manager, EMEA, 10x Genomics; and Dr. Paul Zajac, Segment Marketing Manager, Cell Biology, Illumina. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. Our speakers will now begin their presentations.
Hello, and thank you for spending time with us this afternoon. Before we get the chance to hear more about the exciting research from Bernard, I will give you a brief introduction to 10x Genomics and how you can transform your understanding of genomics, transcriptomics, and epigenomics. As you all know, human biology is immensely complex. From the trillions of cells and types in the human body to the genes and nucleotides found in the DNA of those cells, to the structure of the chromatin, to the translation of genes which differ per cell type. The complexity is enormous, yet thanks to your work, there are many knowns, but still so much left to discover. And as an example, defining this complexity has been taken on by the Human Cell Atlas Global Consortium. And much like the Human Genome Project fundamentally changed the way we explore biology more than a decade ago, the Human Cell Atlas will again change the way that we explore biology, ultimately to influence the treatment of disease. It is 10x's mission to accelerate these studies and reveal these complexities through the solutions we offer. And the core technology that drives the scalability and ease of use of our product is a microfluidic system for massive throughput and molecular barcoding and partitioning of reagents into nanoliter-sized reaction vessels. The 10x portfolio started out with our first implementation of the GemCode fluidics technology with linked reads for genome and exome sequencing and de novo assembly. We then extended the technology for single-cell capabilities with single-cell gene expression, single-cell immune profiling solutions, and innovating on our technology for single-cell CNV solution. Our newly launched set of products include the single-cell attack solution for analysis of open chromatin in epigenetics. Also the implementation of feature barcoding technology, which allows simultaneous measurements alongside the single-cell gene expression and single-cell immune profiling solutions in the same cells. And our newest announcement, the availability of a substantially improved single-cell gene expression solution. So let's take a closer look at these different offerings. So gene expression analysis is a standard and robust tool for understanding the transcriptome, but traditional bulk RNA-seq methods really just give you singular data points per sample. With the scale and ease of 10x, you can profile tens of thousands of cells and multiple samples at a time and beautifully see the clusters and subclusters of cell types. To the right here is a two-dimensional view of a T-SNE plot where each dot is a cell clustered based on multidimensional information and its similarity, then color-coded by known cell markers. We then took the single-cell gene expression functionality and applied it for the study of the adaptive immune system. Here, you can not only identify the subtypes of immune cells based on gene expression profiles, but also in T cells, characterize pair alpha and beta chain receptor sequences, or in B cells, identify paired heavy and light chain immunoglobulin sequences, giving BDJ recombination insights and clonotype information, all in the same cell. Oh, and we can't forget about one of our favorite model systems, the mouse, which we added to the portfolio earlier this year. And also earlier this year, we innovated on our single-cell RNA solutions and applied them to DNA. Our first application with this technology allows you to resolve copy number events at the single-cell resolution, revealing CNV differences in heterogeneous samples, which are really hard to resolve even with the most advanced informatics in bulk CNV sequencing analysis or in karyotype arrays. So in this picture here, a breast tumor sample was dissected and run through the single-cell CNV solution. And every line in the heat map represents a cell, and the coloring means a deviation from normal diploid cells. So you can see that there is variation within each portion of the tumor and within it. Further, on the top sample, there is a small percent of non-normal cells, which wouldn't be able to be seen in bulk CNV analysis. Although this has been applied with a cancer sample, it is also a tool that can be used to look at mosaicism at the single-cell level. The single-cell attack solution allows for interrogation of open chromatin sites in single nuclei for a more resolved understanding of gene regulatory mechanisms and epigenetics. Here we want to show this single-cell resolution of attack to the well-established bulk method. And these are representative tracks of open chromatin regions from the GM128-78 lymphoblastoid cell line, and this portion of the genome represents a known region that has open chromatin sites. The upper orange track represents data gathered from 50,000 nuclei in bulk, while the lower green track is aggregated from about 600 individual nuclei run on the chromium single-cell attack solution. So even with data from a 75-fold fewer nuclei, the single-cell attack solution demonstrates a similar sensitivity to that of bulk ATAC sequencing. And the lower box shows the open chromatin sites, but at single-cell resolution from the same sample as in the aggregate green above. And the next capability that is now available for ordering is the feature barcoding technology with our immune profiling solutions. And the applications that we've started with are the capability to map TCR clonotypes to multiplex sets of peptide MHC molecules, which are antigen presenting molecules, and this is to understand the T cell specificity to the cellular level. In the picture to the left, you'll see a T cell that has specificity for a certain antigen and has a peptide MHC dextramer from Immudex bound to it. We then put those labeled cells through the chromium immune profiling solution and are able to generate profiles that show the clonotype specific to the antigens presented. And in addition to having the feature barcode capability for our immune profiling solution, we also now have it available for our single-cell gene expression solution. And the applications that can be enabled through this are simultaneous measurements of gene expression and cell surface proteins on the same cell, but also highly parallel functional genomics with simultaneous gene expression and CRISPR screening. With this feature barcode capability, you can actually measure up to 4 different readouts in the same cell. So 1, gene expression of the cells, 2, TCR and Ig paired receptor sequences, 3, T cell specificity to antigens, and 4, cell surface proteins. So this is a huge step towards true multi-omics approaches. The newest announcement from 10x is how we push the boundaries of our single-cell gene expression solution with improvements in chemistry and consumables for up to 2-fold increase in gene sensitivity. In this set of data, our version 2 and version 3 gene expression solution was run with a complex cell type, the embryonic mouse heart. You can see in the chart that the gene sensitivity is 2-fold greater at the standard reads per cell. And this improvement in gene sensitivity gives greater experimental design flexibility and allows to detect more genes per cell without the need for additional sequencing reads. Or one could achieve the same cell and gene metrics today with up to half the amount of sequencing reads, which means cost savings, or sequence even more to maximize the number of genes detected. All of these solutions are available for order and consist of our Chromium controller, reagents and consumables, and easy-to-use pipeline and visualization tools, making it as straightforward for anyone to incorporate these methods into their experimental goals. We hope that when you're thinking about the understanding of genomics, transcriptomics, and epigenomics in single cells and beyond, you know that 10x Genomics solutions are there to help and answer those questions. And with that, I would like to thank you very much for your attention. And now, Pavel, I'll hand over to you.
So thank you very much, Annika, for the very nice presentation about 10x Genomics. And thank you, Alexis, for the very nice introduction to this webinar. And of course, the biggest thanks goes out to all of you attending this webinar and spending time with us. So single cell is a field that's really close to my heart, because before joining Illumina around 5 years ago, I used to work with single cell RNA sequencing. And it's tremendous to see just how much this field has evolved over the last couple of years. In this short presentation, I wanted to briefly introduce how our sequencing systems enable you to do a wide range of single-cell sequencing applications. And as this presentation will be very short, if you have any questions, any particular project you are planning, you want to evaluate which sequencing options that are available, please don't hesitate to get in touch with us. We are more than happy to assist. So why run single cells? And this is probably something that's familiar to most of you, but I wanted to go through this short example just to make sure that we are all on the same page. So here you see two collections of cells, A and B. And if you treat these cells in a bulk fashion, you see that the total number of this red transcript is 6 in both cases. So looking at it at the bulk level, you might come to the conclusion that case A and case B are exactly the same. But digging deeper, having the single cell resolution, you'll see that in case A, all cells express this red transcript, whereas in case B, only a few cells express it to a very high level, whereas most of the cells are silent. So by having single cell resolution, you're able to tease case A and case B apart. And as mentioned, there have been tremendous advances over the last couple of years. And we are also seeing a tremendous increase in the number of publications where single cell sequencing has been used. So it's being widely used across many different areas, from cancer research to immunology, from stem cell research to neurobiology. And I'm really looking forward to hearing the presentation by Assistant Professor Thienpont about how he has used this exciting new tool. But it is important to acknowledge that single cell is not a one-size-fits-all application. There are, of course, different study aims, different tissues, different organs, and different biological questions that require different numbers of cells. And of course, there are also different modalities to be studied. For example, some cases you might want to study RNA, other cases DNA, maybe proteins. And we are seeing that there is an avalanche of new methods becoming available, allowing you to study more and more facets of biology at the single cell level. And there is also a drive towards multi-omics, to study multiple modalities or multiple levels of the central dogma in individual single cells in a very high throughput manner. And one example is the site-seq or the REAP-seq type of methods with which you can study both the expression of RNA and protein simultaneously. But no matter what you are interested in studying, we do have a sequencing system, a sequencing solution that matches your output needs. So we have systems from the iSeq 100 instrument giving you 4 million reads, all the way up to the NovaSeq 6000 instrument giving you 20 billion reads. And we strive for our systems to be as flexible as possible. So imagine that you want to do a single cell deep characterization study. Maybe you want to take a look at 1,000 cells of a particular cell type and sequence them at a quite high depth of 500,000 reads per cell to get an understanding of them in more detail. So the total sequencing output required here is 500 million reads, which is an excellent fit for the S prime flow cell for the NovaSeq, so the smallest flow cell for the NovaSeq system. But now imagine that you also want to do an atlasing study where you take 200,000 cells from a particular tissue, from a particular organ, to understand the cellular composition of that tissue, of that organ. And you want to profile those cells at 50,000 reads per cell. The total output required here is 10 billion reads, which is an excellent fit for the S4 flow cell for the NovaSeq system, so the largest flow cell available. So simply by selecting the most appropriate flow cell, you are able to use the NovaSeq system for a number of different applications. And I also wanted to highlight our iSeq 100 system. So this is, as mentioned, our smallest sequencing system. And as you can see, it gives you 4 million of reads per run. And this is too limited for most single cell applications, but it's an excellent fit for library quality control. So you could quality control your single cell library on the iSeq 100 system before running it on, for example, NovaSeq system. And in addition to the flexibility, we continuously strive to evolve our sequencing systems to enable you to do more using the existing systems. These are examples of technological advances announced at the American Society of Human Genetics annual meeting in October 2018. So we demonstrated 2 times 250 runs on both the iSeq and the NovaSeq systems, extending the platform capabilities and the potential applications. And perhaps of more interest for the single cell field, we released a new version of the NextSeq reagent kits. And these have improved robustness and flow cell stability. So I wanted to once again reiterate that Illumina systems enable a wide range of single cell sequencing applications. So if you have a particular project in mind, if you have any questions, if you want to find out which is the optimal system and solution for your needs, please don't hesitate to get in touch. As mentioned, we are more than happy to assist. And of course, a big thank you for joining us today.
So hello everybody, I'm very happy to talk to you today about the phenotype molding of stromal cells in the lung tumor microenvironment. And I'm also very grateful to 10x and Illumina for organizing this webinar. Just a very quick disclaimer that the opinions expressed here are mine and not those of Illumina. So today we are going to talk about the lung tumor microenvironment. And as many of you listening will probably know, lung tumors do not only consist of tumor cells, they also contain fibroblasts that provide support to these cancer cells. There's endothelial cells that line the blood vessels that supply nutrients and oxygen to the tumor. And then there's a whole host of immune cells that are attracted to tumors. And all of this happens in the context of a normal lung. So you have also all the different alveolar cells that are present normally in the lung that can also be found in the lung tumor microenvironment. So all in all, this makes up a very complex cellular ecosystem. And this is not something that is purely out of scientific interest. This is also therapeutically very important, as many of these cell types are established targets for cancer therapies. For example, T cells are the target of immunotherapy, and endothelial cells are the target of anti-angiogenic therapies. But the question we were asking is whether these are indeed all the cell types that are present and whether they form uniform populations. And finally, also how they are affected by the tumor microenvironment. And to study this was until recently very difficult, but then came along 10x and single-cell RNA sequencing, and this allowed us to study the diversity and the heterogeneity of the tumor microenvironment in a lot more detail. So to do this, very briefly, we isolated tumor samples as they came from the clinic. We added proteinase and collagenases to them to digest away the extracellular matrix. And this provided us with a single cell suspension that we then took forward to make libraries out of using the 10x technologies. A very brief overview of the study that we've done. So these were all treatment-naive patients with non-metastatic non-small cell lung cancer. We focused our analysis on 5 different patients that underwent primary lobe resection for curative intent. All of these patients had either non-small cell lung cancer of the squamous or the adenoid subtype, and some of them also suffered from chronic obstructive pulmonary disease. All were former smokers. From these patients, we isolated 19 samples in total. So 4 came from the normal lung lobe, and 15 came from the tumor. So from every tumor, we isolated 3 different samples for reasons which I will not discuss in detail further during the current presentation. All in all, from all these samples, we isolated a little over 50,000 cells, and this allowed us to sequence the transcriptome of these 50,000 cells and produce a little over 3 billion unique transcripts, which we then took forward for analysis. And to analyze these, and let me just briefly explain to you how this is typically done. I know that a lot of you that are listening will know this, but transcriptomes of single cells are analyzed or are depicted at least often using TSNE plots, so T-distributed stochastic neighbor embedding plots, in which every dot represents a cell, and the closer different cells are together, the more similar they are in terms of gene expression pattern. So when we look at established marker genes, we can identify, for example, alveolar cells on your left, endothelial cells, epithelial cells, fibroblasts, which are collagen positive, B cells, myeloid cells, T cells, and apart from those, of course, also the cancer cells. Now, before we dove into these analyses, we first wanted to do a little QC, and for this we compared the classical methods to do RNA sequencing. This is bulk RNA sequencing with our single cell transcriptome. So we collated all single cells from one sample and compared the expression of those collated cells to the bulk transcriptome. And this revealed that for some marker genes, such as COL1A1, there was an enrichment in the bulk, whereas other marker genes showed a depletion in the bulk and enrichment in the single cell RNA sequencing. This was something that was not only evident from the these single marker genes, also when we did gene ontology analysis for genes that were typically downregulated in the single cell data set, or genes that were typically upregulated in the single cell data set, we observed similar patterns. So genes that were downregulated were often enriched in extracellular matrix proteins, whereas genes that were upregulated were very often involved, for example, in T cell activation. We also quantified the number of cells in bulk mRNA sequencing, or we estimated these based on the marker genes that we identified, and this also showed us the same pattern, namely that T cells are enriched and B cells are enriched and myeloid cells tend to be enriched in the single cell RNA sequencing dataset, whereas alveolar cells and cancer cells and endothelial cells and fibroblasts in particular tend to be depleted from the single cell RNA sequencing. And this probably simply reflects a difference in dissociation efficiency. So some cell types, such as fibroblasts, are much more difficult to dissociate when compared to T cells, and so probably this is why they are underrepresented in the single cell RNA sequencing. That said, overall the expression pattern was very similar between bulk and single cell, so we were happy to proceed and to continue with our analyses. And this was done using SEURAT. We tested a number of different analysis platforms that are out there. We tried SE3, RCA, CIDR, and also Scenic. And out of these, when we analyzed it, when we looked at quality metric, the NMI, we saw that SEURAT in general produced the most consistent results out of all the platforms, although SE3 was also quite good, as you can appreciate from the plot on your lower left. So we continued with SEURAT. And within SEURAT, when we performed clustering of our cells, we always used a resolution that was quite conservative. And the resolution and all the other clustering parameters that we used were chosen so that they were robust to changes in the parameter settings, so that the results that we obtained were actually quite robust. When we now go back to the TSNE plot that I discussed, that I showed to you earlier, and we color code every cell according to the sample of origin, you can already appreciate that for some of these clusters, and I highlighted the cluster names for you on the right. So for some of these clusters, you can appreciate that they are predominantly tumor-derived, and this goes obviously for cancer cells, but also, for example, for B cells, and to a lesser extent for fibroblasts, whereas other clusters are predominantly non-malignant tissue-derived, and this is, for example, true for the alveolar cells. When we now look at the panel in the middle, where we have color-coded every cell according to which patient the cell comes from, you can appreciate that most of the stromal cell types are actually derived from multiple patients, so they share transcriptomes across different tumors. In contrast, the cancer cells tend to be patient-specific, so we have one cluster that originates from patient 5, one cluster that originates from patient 4, 1, 2, and 3. So in conclusion, cancer cells tend to be predominantly tumor-derived and they cluster per patient, whereas other cell clusters tend to be more mixed in terms of sample and patient. And as I mentioned to you earlier already, the recovery of these cells tends to be cell type dependent. Next, we wanted to know how homogeneous these different stromal cell types are and how they are affected by being in the tumor microenvironment. And so to do this, we analyzed each cell type again, but independently. So to illustrate this a bit more clearly, we take all 1,592 endothelial cells, which are Claudin 5 positive, marked in red on the left, and we recluster them. And this allows us to ask, which expression patterns allow us to distinguish different types of endothelial cells, and do they actually cluster separately? The answer is, of course, yes. Otherwise, we wouldn't be sitting here. Our analysis revealed a strong influence of the tumor microenvironment in that normal endothelial cells and tumor endothelial cells tend to cluster separately. So this is, as can be appreciated on the right. Overall, we identified 6 different clusters of endothelial cells. 2 clusters were mainly normal tissue-derived. 2 clusters were mainly tumor-derived. There was one mixed cluster that was mainly composed of low-quality endothelial cells, and then cluster 6, which is also derived of 2 different sample origins. Now, when we look at marker genes that are known to mark different types of endothelial cells, we can already tell that cluster 6 actually corresponds to lymphatic endothelial cells because these are podoplanin positive, whereas all the other clusters are marked by FLT1, and this is a marker of endothelial cells that normally line the blood vessels, so these are blood endothelial cells. Now, because we have the transcriptomes of endothelial cells that are derived from non-malignant tissue and from tumor tissue, we can specifically look for marker genes that mark either normal endothelial cells or tumor endothelial cells, and I've highlighted 2 of those for you on this slide. So MT2A is a marker of normal endothelial cells that we identified, and HSPG2 specifically marks tumor endothelial cells. These are just two of the markers that we identified, but we've identified over a dozen for each of these. And this allowed us to turn to bulk RNA sequencing data that was characterized in the Cancer Genome Atlas. And here we're looking at the aggregate of about 1,300 samples that either were derived from lung, from squamous tumors or adenoid tumors. And as you can appreciate, normal endothelial cell markers tend to show a higher expression in normal lung tissue when compared to tumors, whereas tumor endothelial cell markers tend to show a higher expression in tumors than in the normal lung. So this also validates a bit the marker genes that we identify. We've also used immunofluorescence to see whether we can indeed identify specific subpopulations of endothelial cells as being marked by one of these new marker genes. And here we are showing, for example, results for ACKR1, which is a marker of endothelial cell cluster 3. This is a tumor endothelial cell cluster. And as you can appreciate on the top, ACKR1 only marks a subset of the cells that are Claudin 5 positive. So Claudin 5 is marked in red here. Whereas if you look at the lower panels for the non-malignant immunofluorescence, there again you can see that ACKR1 marks only a subset of the endothelial cells, but there's a lot more endothelial cells in the non-malignant sample when compared to the tumor sample. So this is something that is also shown in the quantification, which is shown at the bottom. So there's a lot more Claudin 5 positive cells in the normal sample when compared to the tumor samples, but the fraction of endothelial cells that is actually ACKR1 positive. And so ACKR1 is a marker for cluster 3, a tumor endothelial cell marker. So the fraction of these endothelial cells is much higher in tumor samples than in normal samples. And obviously identifying marker genes is all very interesting, but we wanted to understand the phenotypes of and the differences in phenotype between normal and tumor endothelial cells. And to look at the phenotypes of cells, we scored their expression using 50 hallmark pathways. So the hallmark pathways, these are sets of genes described in the molecular signature database from the Broad, and that are generally agreed upon as being important in cell biology. Each of these hallmark pathway gene sets were next scored using gene set variation analysis in every single cell, and this produces a picture that looks somewhat like this. So the more blue a cell is, the higher the expression of, for example, the MYC target pathway is. Now, I'm not showing you the MYC target pathway here just by coincidence. Indeed, when you look at the MYC targets, this is the pathway that shows the strongest enrichment in tumor versus normal. So all the cells which are on your upper left, these are in fact tumor endothelial cells. The cells which are on your lower right, these are normal endothelial cells. And as you can appreciate, there's a lot more MYC target pathway activity in tumor endothelial cells than in normal endothelial cells. In line with this, also when we look at MYC expression on its own, we see that this is much higher in tumor endothelial cells than in normal endothelial cells. Now, MYC is a very interesting protein, and it's been studied a lot, and one of the things it classically does is that it gives metabolic changes to cells that feed back to global RNA production. So in other words, cells that have a high MYC expression tend to have a lot more RNA production than cells that have a low MYC expression. Now, because we do 10x, and with 10x we can identify unique transcripts, and we can count those unique transcripts in each and every single cell. This allowed us to look at the phenotypic outputs of increased MYC expression, namely how many unique transcripts can we actually identify in each cell. And again, in line with an increase in MYC expression in tumor endothelial cells, we see that we detect a lot more unique transcripts in tumor endothelial cells than in normal endothelial cells, even though samples were sequenced at a comparable depth. And in other words, tumor endothelial cells are indeed transcriptionally more active. All of this suggests vasculogenesis may be sensitive to MYC inhibition, and there's at least some hints in the literature to support this notion. Indeed, it has been shown quite a while ago already that MYC is essential for vasculogenesis and angiogenesis, for example, during development. When we now go back to our 50 hallmark pathways and we look all the way at the bottom, so which is the pathway that is most downregulated in tumor endothelial cells, we noticed something very remarkable, namely that the inflammatory response was most downregulated in tumor endothelial cells. Why do I say this is remarkable? Well, because tumors tend to be very inflammatory. When we look more in depth at which genes are actually downregulated, we note that a lot of these genes are involved in antigen presentation and in immune cell homing. So we have, for example, ICAM1, which is a protein that is expressed on endothelial cells and that allows immune cells to adhere to the endothelial cells that line the blood vessels and subsequently to penetrate the tissue and deal with the inflammation as it is present. To understand this a bit more, it's important to realize that your blood vessels actually form the primary interface between the tissues that they perfuse and the immune system as it is going through your bloodstream. And when there is inflammation in a tissue, this inflammatory signal is typically relayed through endothelial cells that line the blood vessel, and they then present signals to the immune system that allow the immune system to recognize that there is inflammation and to penetrate the tissue. So all these data suggest that this rewiring of tumor endothelial cells contributes to the immunosuppressive phenotype of tumors, and that if we can make tumor endothelial cells more normal, that this may help with immunotherapy. And there's at least some suggestions that combining anti-angiogenic therapy and immunotherapy, that this might actually be a very successful strategy. And also in line with this is this seminal paper that was published now a decade ago, which showed that vascular normalization, in fact, it promotes immune destruction. So when we make tumor endothelial cells more like normal endothelial cells, this actually promotes immune destruction of tumors. Now, to understand why these tumor endothelial cells show such a divergent transcriptome from normal endothelial cells, we turned to CNIC, and this is a new method that was developed by some very clever people down the corridor here from the lab of Stijn Aerts. And what CNIC actually does is, in a first step, it looks for genes which are co-regulated. So you have here genes which are either always upregulated in a cell or either always downregulated in a cell. And then the very clever trick is CNIC looks for conserved transcription factor binding sites or conserved motifs upstream of genes that tend to be co-regulated, and it looks for specifically for those motifs that are present at each of these genes in this set of co-regulated genes. CNIC also looks for the transcription factor that recognizes this conserved motif, and in that way it can build a regulon. So this is a set of genes and a transcription factor that are all co-regulated, and we can score each cell for the activity of this regulon, and then in a last phase, contrast cells that have a high regulon expression versus cells that have a low regulon expression. And when we do this for normal and tumor endothelial cells, we can identify different transcription factors that are either active in one or in the other. And the most striking here is the FOS-JUN pathway, which appear to be very strongly downregulated in tumor endothelial cells. And why is this an interesting pathway? Well, because from the literature we know that FOS and JUN can actually activate ICAM1. So this is the protein that was involved in immune cell homing. So when we lose FOS and JUN, we in turn lose ICAM, and this suggests that loss of FOS and JUN may underlie the immunosuppressive phenotype of tumor endothelial cells. So to summarize, tumor endothelial cells are very distinct from normal endothelial cells. They upregulate transcription through MYC activation. They downregulate immune cell attraction pathways, and they show a decreased FOS and JUN activity. So apart from endothelial cells, there's also a lot of other stromal cells that we've looked at in this study. And let me just briefly mention to you what we've found in the T cells, because this is also quite interesting. So we've looked at almost 25,000 T cells. T cells make up by far the most abundant subpopulation that we could isolate. And when we cluster these T cells, we find actually 9 different clusters of T cells. Most of these are strongly enriched in the tumor microenvironment, as you can appreciate from this slide. And when we look at known marker genes, we can tell that we have identified natural killer cells, CD8 positive T cells, CD4, regulatory T cells, exhausted T cells, which are a subpopulation of the CD8 positive T cells. And then finally, there's the proliferating T cells, which are on your lower right. So are these all the T cell subtypes that we could identify? And what about other known T cell types? Well, when we look at, for example, cluster 6, so this is the cluster which is on top there, and when we look at the overplotting density, so how many cells are in every quadrant on the plot, we see that in fact cluster 6 probably consists of 2 different clusters. And also when we look at CD3 expression, we see that only one of these clusters is marked by CD3. So the upper one of these clusters actually corresponds to NK cells and not NKT cells. So why are these 2 clusters then clustered together? Well, quite simply, probably because both are NK cells, so they execute the same functions and they share the vast majority of their transcriptome. This result also highlights that we here cluster cells by function and not simply by their lineage. As you all know, NK cells are part of the innate immune system, whereas NKT cells are part of the adaptive immune system. Apart from these, we also identified ILC1-like cells and gamma delta T cells, which are part of CD8 T cell cluster 2. And overall, the fact that we combine known cell types within one cluster strongly argues against that we did over-clustering and that we artificially generated new cell types. So can we also identify new T cell types? Well, there's at least one that I thought was very interesting, namely this subpopulation of CD8 T cells that doesn't express granzyme B, but that does express granzyme K. While there are some hints in the literature to suggest that these are immature CD8 T cells, I think there's a lot more work to be done to understand the exact biology of these subcluster. And likewise, there are CD4 T cells that express CXCL13, of which the function has been hinted at in the literature, but that is for the moment still quite unclear. When we look at the marker genes and quantify their expression in bulk RNA sequencing data from tcGA, we can again recapitulate the different distribution of these clusters between normal and tumor cells. And when we look at the pathway activity of each of these, and this is shown here on the right, I would say that by and large, being in the tumor microenvironment has a similar effect on each of these subpopulations of T cells. So when we look at the individual clusters of CD8 T cells, it is very clear that there are striking differences between those that are derived from the tumor and those that come from the normal tissue. So cluster 8, for example, shows a very strong increased proliferation when it is in the tumor, whereas clusters 4 and 8 show a strong increase in interferon signaling and in allograft rejection. The allograft rejection pathway is quite interesting because it was composed of genes that are typically upregulated when there is an organ transplant that is being rejected. So in the context of a tissue, this probably corresponds to neoantigens that are presented specifically in the tumor and that do not correspond to the normal genetic makeup of the tumor host. In line with this, clusters 4 and 8 also upregulate strongly checkpoint molecules. So we have PD1, CD8, CTLA4, and LAG3, which are all upregulated in clusters 4 and 8. And this is together with a strong upregulation of T cell activity, as shown by the upregulation of granzyme expression. So what this actually allowed us to do was to correlate T cell activity as measured by the activity of granzymes with all other genes that are specifically expressed in T cells. And what this revealed is that actually all the known checkpoints that are either accepted as therapeutic targets or that in clinical trials are actually very strongly correlating with granzyme expression across these 25,000 T cells. And apart from these, there's also a lot of new potential targets that we propose are potentially also new targets for immunotherapy. So in conclusion, we identify all the known and some perhaps unknown or less well characterized T cell subtypes. Most T cells activate similar pathways in the tumor microenvironment. Subclusters of CD8 positive T cells clearly have different pathways which are active depending on whether or not they are part of the tumor microenvironment. And known as well as novel candidate checkpoint molecules correlate with CD8 positive T cell activity. So finally, let me just briefly show you some of the data that we obtained for fibroblasts, because we were quite excited when we saw this. Fibroblasts are typically considered to be a quite uniform cell type, and they're very difficult to study in isolation, in part because they tend to adopt a plastic adherent phenotype when you try to culture them. So when we look in vivo, what expression patterns actually distinguish these different types of fibroblasts, we saw in fact that we could identify 7 different clusters of fibroblasts. These are mostly enriched in the tumor microenvironment, but they show a remarkable phenotypic diversity. And this was particularly true when we looked at the expression of extracellular matrix molecules and collagens in particular. So as you can appreciate from the panel on the right, while most of the clusters expressed collagen 6, cluster 2 specifically expressed collagen 4, cluster 5 specifically expressed collagen 5, cluster 7 specifically expressed collagen 8, etc., etc. So each of these fibroblast types apparently expresses its own unique extracellular matrix repertoire. And overall, we move from a situation where a little over a dozen different cell types in the lung tumor microenvironment are recognized to this situation where we see 52 different types of cell types. And importantly, this was not limited to the 5 patients that we studied, because when we tried to replicate this in 3 additional samples, we could recover 45 of these 52 different stromal cell subtypes. Now, because we have marker genes for each of these, we can also look at how they affect tumor biology. And what we, for example, very interestingly noticed was that they are strongly anti-correlated. Their presence is strongly anti-correlated with mutational burden. Now, here we don't think this is actually part of the normal biology. What we think is that this actually reflects errors in mutation calling, where a higher stromal cell admixture leads to a lower allele frequency and more difficulties to identify mutations. Very interestingly, stromal cell abundance was also correlated to patient survival. In particular, in squamous tumors, we noted that high abundance of stromal cells was detrimental for patient survival. And so, for example, when we look at certain types of epithelial cells or alveolar cells, we noted that higher abundance of these cells was associated with poorer survival. And this was observed in TCGA data and could be replicated in 2 additional studies. So to summarize, I generated the first comprehensive catalog of the lung tumor microenvironment in which we detected 52 different cell subtypes and observed, for example, a striking fibroblast heterogeneity. We saw a remarkable influence of the tumor microenvironment on endothelial cells and also on myeloid cells. These are data which I hadn't had time to discuss to you today. And we identified specific marker genes that can serve as biomarkers for stromal therapies. We also identified specific pathways and transcription factors that are active in the tumor microenvironment and that may represent new therapeutic vulnerabilities. And then finally, the relative abundance of cell subtypes is correlated with patient outcome. So if you want to learn more about this, all of this was published recently in Nature Medicine. Within this publication, you can also find the link to our website on which we have put all our data sets available, and you can upload them from there into Scope, which is a graphical user interface that allows you to interactively explore the data that we have generated here. And then finally, you can also come to a conference that we are organizing next year in March, where leaders in the field, as well as people from, for example, 10x and Illumina, will come and discuss the next hot topics in next generation sequencing. So with that, I would like to end. Let me just briefly still acknowledge everybody that contributed to this study, and in particular Dieter Lambrechts from the Flemish Institute of Biotechnology, Steinhardt and Sarah, who contributed scenic, and everybody from the hospital that contributed samples. And of course, all of you for your attention. Thank you.
Thank you, Bernard, Annika, and Dr. Zajac for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, what is the most robust way to normalize read numbers between single cells? We observe huge differences in housekeeping gene expression, and mitochondrial transcripts seem to be influenced by our treatment.
Hi. So the way we normalized read numbers between single cells is just initially a very classical normalization to the total read count within that single cell. The fact that you see huge differences in housekeeping gene expression is not something that we typically see. What I do know is that mitochondrial transcripts tend to change in expression levels as cells undergo apoptosis, because then these mitochondrial transcripts tend to get more prevalent. So if you see a high level of mitochondrial transcripts, then this could perhaps signal that there's an increase in apoptosis of your cells. You can normalize out, for example, increased mitochondrial transcript expression, or normalize out batch effects using classical models, which are implemented also in SERAT. So maybe that's something that you could try to overcome this. The reason why you see these differences is probably in part technical and probably in part biological. So it's definitely something to take care of and to look out for.
Now, our next question is this data published yet?
So there the answer is yes. As I mentioned in the presentation, all this data is published in Nature Medicine a couple of months ago. And as I also mentioned, you can go to our website, download all the analyzed data, or you can get in touch if you really want to get the gene expression matrices directly. And you can also go to Scope, where you can interactively play with all the data, and it's something that, in my experience, people find really quite handy, and people tend to really enjoy doing so. If there's any particular cell type that you're interested in, or any particular gene or set of genes that you want to explore, then I would suggest that you go to Scope and just see for yourself.
Now, what was the depth of sequencing per cell?
So this is something that is highly variable, in fact. So we tend to aim for quite a good sequencing depth. Let me see if I can recall the numbers off the top of my head. I think we went to about let's say 100 million reads per 1,000 cells, or 50 million to 100 million reads per 1,000 cells. Again, this is something that is highly variable. Also, not just because the number of reads per cell tends to be very variable, not just because of how much sequencing effort you put into it, but also because the number of reads that you recover from every cell varies tremendously, both for technical reasons. I don't know, maybe less transcripts are captured in one compared to the other, let's say, microwell. And on the other hand, there's also simple biological differences between different cell types, and some cell types just on average express less transcripts than others, and cancer cells, for example, tend to show very high transcript numbers. Whereas, for example, T cells show on average a bit lower transcript numbers. So there's no simple answer to the question, but that's I guess as much as I can meaningfully say about that.
Now let's move on to our next question. How fixed are these cellular phenotypes? Are these truly cell types, or do they represent cell states?
No, that's a question that I get quite frequently, and it's for sure it's at least in part a matter of semantics. What do you call a cell type and what do you call a cell state? I think it depends a bit on what domain you are working in. If you're working in immunology, then a cell type is something else than when you're working in embryology, for example, or when you're working in neurology. Cell types tend to be very much defined by function, whereas in other domains, cell types tend to be defined by for example, by morphology. So whether they are really cell types or cell states, that's something that I think the jury is out there now for the moment. I cannot say for sure whether they are cell types or cell states, whether they are interconvertible or not. It's something that I think is a very interesting question, and something that we are definitely very keen to explore further in future studies.
We are getting some great questions in today, so let's move on with a couple more. Could you give some details about scenic? Can we use it with bulk RNA-seq data or only single-cell RNA-seq data?
So scenic is something that is specifically designed for single cell data because it really needs a lot of independent signals to independent, let's say, samples, and every single cell for scenic is one sample. So it needs a lot of different samples
to identify sets of co-regulated genes. So these are sets of genes which tend to be either up or down in a subset of your cells. That's genes which are highly co-regulated. So I don't think you can use this with bulk RNA-seq data. Another tool that was developed and that in my hands at least works very well is called ISIS-Target. It was developed by the same lab that also developed CNIC, so the lab of Steenaert. And ISIS-Target similarly looks for enriched transcription factor binding motifs upstream of sets of co-regulated genes. It just applies a bit more of a, if I remember correctly, it applies a bit more of a stringent threshold on what is considered a conserved motif. One of the very neat things about CNIC, and this is something that you cannot directly do with ISIS-Target, is because in CNIC you also have the differential expression of the transcription factor itself. You can directly link the expression of that transcription factor to the increased activity of genes that all share upstream the motif that normally is the target for this transcription factor. So what CNIC does really a great job at connecting the transcription factor to the transcription factor binding motif that is upstream of your set of co-regulated genes.
And it does look like we have time for one more question. Do you have any insight in the spatial organization of these cells? Where are they in the tumor with regards to each other?
So we have some insight. I know that people are developing ways to get a lot higher resolution insight in these, for example, with spatial transcriptomics technologies or with FISSEQ. We don't have those data at hand for us. What we did do, I mentioned this early on in the presentation, that for every tumor we isolated 3 different samples. And in fact, one of these 3 samples originated from the tumor core. Another of these 3 samples came from the edge of the tumor, and one was in between. So this allowed us to get a feel for how the distribution of different cells is within the tumor. And this clearly showed that, as one would expect, that tumor cells, cancer cells, that they tend to be enriched in the tumor core and that they are a bit depleted from the tumor edge, and vice versa, that many of the stromal cells, but clearly not all, and if you want to get more information on this, I suggest you go to the paper, but that many stromal cells, and in particular alveolar cells, tend to be enriched at the edge of the tumor. Likewise, we could see that cancer cells that are at the tumor core tend to be more hypoxic than cancer cells which are at the edge of the tumor. And again, this is something which is pretty much in line with what is described in the literature. So we have a rough idea about where these different cell types are, but if we want to get a more fine-grained information on how these different cell types are, let's say, juxtaposed, and whether cell type A that we describe is associated with cell type B or whether it's associated with cell type C, then you need to go either to spatial methods or you have to rely on the recently described co-expression of the agonist receptor interactions. I believe there was a paper in Cell that was published a couple of months ago that looked at the development of basophils in the lung. It was an excellent paper, and if this is something that you're truly interested in, then I suggest you go and have a look at that one.
Thank you again, Bernard, Annika, and Dr. Zajac for your time today and your important research. We would also like to thank LabRoots and our sponsors, Illumina and 10x Genomics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information that you provided at the time of registration. This webcast can be viewed on demand through May of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.